Hi, my name is Jocelyn Pierre-Louis, and thanks for stopping by my poster. I am excited to share how ionizing radiation causes pulmonary fibrosis to shift their metabolism to glycolysis. Exposure of the lung to ionizing radiation, such as during radiotherapy for cancer, can result in pulmonary fibrosis, which has few treatment options, and there is no standing cure for this disease. Radiation-induced pulmonary fibrosis is characterized by an accumulation of extracellular matrix proteins produced by scar-forming cells that form scar tissue. This results in disruption of gas exchange and even death. The Sign Lab and others have shown that metabolic reprogramming is a hallmark of idiopathic pulmonary fibrosis. In normal lung tissue, we have this process called glycolysis, where glucose, a sugar molecule, is broken down into pyruvate. Pyruvate then shuttles into the mitochondria to undergo this process called oxidative phosphorylation that gives us energy in the form of ATP. In IPF lung tissue or lung tissue treated with transforming growth factor beta, a profibrotic signaling cytokine, we see that pyruvate doesn't go into oxidative phosphorylation, but rather is converted to lactate via this enzyme called lactate dehydrogenase. Lactate then shuttles out of the cell into the extracellular matrix to then bring this activation of TGF beta in a pH-dependent manner. So we see this happening in IPF. This project hypothesized that ionizing radiation would cause aerobic glycolytic metabolic dysregulation in primary human lung fibroblasts. So human lung fibroblasts from non-fibrotic patient donors were cultured and irradiated with varying doses of radiation or treated with TGF beta. We see here, this is an immunohistochemistry staining where we're looking at protein expression. DAPI is staining the nuclei of the cells in blue. That lets us know that cells are present. We also have alpha smooth muscle actin and collagen 3, which are profibrotic markers. And we see that as we increase with radiation, we see more green staining of ASMA and collagen 3, indicating that the cells are becoming more profibrotic after exposure to radiation. Looking at gene expression of metabolic and profibrotic markers, we see that there's this induction or increase in these markers. Remember, lactate dehydrogenase A is important for that conversion in pyruvate to lactate. Pyruvate dehydrogenase kinase 1, PDK1, is important for, again, shuttling pyruvate to go into lactate. We see that there is a dose-dependent increase with radiation. We also see that the profibrotic marker alpha actin 2 is also increasing with radiation. And this is happening across different patient donors. Looking at protein expression of PDK1, we also are seeing this induction with radiation, where PDK1 expression is increasing across two different donors with radiation. And we can also see that as these bands are becoming more robust and more deep, indication more protein expression of PDK1 with increasing doses of radiation. So though we had previously demonstrated that lactate and LDHA expression do increase post-irradiation, the underlying mechanisms remained unclear. Here we see in this project that the role of precursor glycolytic defects may lead to this phenomenon. This non-fibrotic human lung fibroblasts change their metabolism in response to radiation exposure. And we see that it's shifting towards glycolysis with increases in LDHA and PDK1 gene expression. And not only is it shifting it towards glycolysis, it's shifting it in a way that's making the cells more profibrotic. And we see that with active 2 gene expression and with ASMA and collagen 3 expression. So this gives us a clue on looking further into the mechanism of the different changes in metabolism that might also play into why we're seeing these profibrotic changes. Thank you so much for stopping by. Signing off.